Hello everybody, my name is Akhilesh Theory, integrated master and PhD student of SR Lab from Congo University. Today we will going to talk about the introduction to the sensor and the transduction element. In this presentation we will briefly talk about the introduction to the sensor, what the sensor is and the nodes as a sensor, and also we will talk about the sensor and biosensor, some aspect of the sensor that like recognition element, transducer, method of immobilization, performance factor, and area of the application. What are sensor? Do we know biological sensor? We have 5 biological sensor, that is nose to smell, tongue to taste, ear to hear, eye to see, and finger to sense. Likewise, in laboratory, we have the litmus paper for the testing of acid and base. In figure, we can see the litmus paper. When we dip the litmus paper in acidic solution, is changed to the pink, while we do in the alkaline, is changed into blue. So likewise, we have different sensor like physical parameter, and then we have sensor, then we have transducer and observer response. While talking about the classification of the sensor, we already talked about the physical sensor. That physical sensor is like measuring the distance, mass, temperature and pressure. And the chemical sensor, chemical substance by the chemical response. And biological is also types of chemical substance sensor that only doing the sense to our biological means. Now we will talk the nose as a sensor. All human beings have the nose as a sensor for the smell. Figures 1, first figure is the schematic of the sensor. We have some sample, then we have biological detector as a nose, then it transduce, then we have our brain which sense the smell. And the working of biological sensor is like first we have the sample, then we smell through our nose, then nerves direct do processing, and the brain get absorb the response. Sensor and biological sensor definition. Now sensor, actually a sensor device is a device that detects a physical property and record, indicate or respond to it according to the expert dictionary. While talking about the transducer, is also a device that converts and absorbs change into a measurable signal. While actuator is a part of device which produces the display on any types of action. Sensor, we have different types of sensor, as we previously talked, that physical sensor, chemical sensor. Physical sensor that measure the length, weight, temperature, and the chemical sensor is qualitative as well as quantitative, and biological sensor that determine the recognize the biological. And what are the aspects of the sensor? Its recognition element impart the selectivity that enables the sensor to respond selectivity to a particular analyte or group of analytes. It also avoid the interference. Some example like ion selective electrode for the analyzing specific ion, whereas enzymes, nucleic acid are some sensor used in biosensor. Transducer, the detector device. electrochemical transducer. We have voltometric, that is EMF for EMF of the cell at the zero current. Voltmetric is measure the peak current. Conductometric, electrical conductivity of the solution, it measure the electrical conductivity of the solution. And FET based transducer is actually the silicon chip based FET. It is either potentiometric or voltmetric. While talking about the optical transducer, it's absorb fluorescence, means light, that internal reflection, surface plasma spectroscopy, and the light scattering by developing fiber optical. Now talking about the method of immobilization, method to connect the selective element to the transducer. While there are different methods like absorption, microencapsulation, entrapment, covalent attachment, and cross-linking. Absorption is simplest methods, whereas microencapsulation is trapping between the membranes. Same way, cross-linking is a biofunctional agent that is used to bond chemical the transducer to the selective component. Performance factor, they are selective and selective range, and also on the basis of the accuracy, nature of the solution, response time, recovery time, and the working lifetime. Selectivity, the ability to determine between the different substances. As we talk about the litmus paper, that for acid and base it shows the different color, as well as selective range. It can be of few sub millimole up to like 10 power minus 15 more. What is accuracy? Accuracy should be less than 5%. Error should be less than 5%. By nature of the solution, it should measure the pH, temperature, ionic strength. Response time, it should be 30 seconds or more. Recovery time, time that elapses before the sensor is ready to analyze the next sample. Working lifetime, stability is the must. Now we will talk some area of application. We can use sensor as a healthcare. Main area of application, biosensor and the chemical sensor. Measure the blood gas, ion, metabolic. Smart and combinational biosensor simultaneously and continuously measures multiple measurements. In industrial process, we can use to measure the temperature, pH, CO, and O2. While as environment monitoring, potential analytic in the air and water, soil, and nitrate, phosphate, calcium in the soil, and many more. Now come to the chapter 2, transducer element. In this chapter we will talk about the electrochemical transducer, potentiometry, and ion selective electrode. The Knots equation, we also talk about the Knots equation. Then we will talk about the voltmetry and amperometry. We will talk conductivity, field effect transistor, FET, and we will talk some modified electrode. Then at last we will talk about the photometric sensor. Electrochemical transducer. Basically there are three electrochemical processes useful in the transducer for the sensor application. One is potentiometry, voltmetry, and conductometry. Potentiometry and ion selective electrode. First figure is the half shell of a potentiometry, whereas the second figure shows the complete elect complete cell of electrometry. Here you can only in half we only see the solution, electrolyte and the 1 meter, but for the complete there should be the 2 electrode and the solution, and also both solution are connected through the bridge, that is salt bridge. Here we can see the different types of reaction that took place in a complete electrochemical solution. Here we can see the oxidation and the reduction, both are occurring simultaneously. The first figure is only half is Daniell cell, whereas the figure second is half cell of electrochemical. Now reference electrode. The standard hydrogen electrode is a reference electrode to which other electrode may be referred. Some commercially available electrodes are silver chloride electrode, silver and also saturated chamomile. They are the two commercially available electrodes and they are easy for the chemical reaction. Quantitative relationship, the Nernst equation. The basic Nernst equation is the logarithmic relationship derived from fundamental thermodynamic equation. The Nernst equation is R into T logarithm of K. Here you can see the various equation, practical aspect of ion selective electrode. The first figure is the reference electrode combined with another half cell. The second figure is concentration cell. Here we can see the membranes as well as the voltmeter for the half cell reaction. Quantitative relationship, the Nernst equation. Previously also we talked about it is the logarithm relationship derived from the fundamental thermodynamic equation. Ion selective electrode respond to one particular ions more than other. Practical aspect of ion selective electrode. Ionic strength. Keep the constant by adding a high concentration of an indifferent electrolyte, one that does not interfere with the analysis. Now pH electrode, pH control. Maintain a specific pH level, which is practically important for ion sample like fluoride. Interference elimination. Add component to reduce or remove interfering ion. Measurement and calibration. Calibration graph method. We use this when accuracy depends on data point, and this can be also determine unknown concentration. It's also work even with non-Nernstian slope or curvature. Single strand addition method requires only one strand solution. Standard must be close to the expected value. Multiple standard addition method. Combine standard addition with multiple standards. It is more convenient than calibration graph method. Assume a linear relationship and knowledge of a slope. The graphs gives a negative intercept for the result with no additional calculation required. Voltmetry and amperometry. Linearly varying potential between a working and a reference electrode in an electrochemical cell containing a high concentration of electrolyte to make the solution conduct. Linear swap voltmetry. Current through the cell is monitored and plotted in voltmegraph, and this technique is called linear swap voltmetry. In figure, we can see the graph between EMF and current, and this is plotted, and this plotted technique is known as the linear swap voltmetry. Voltmetry and amperometry. Diffusion limited current. The figure you can see they are the various graphs for T1 and T2 and T3. Factors that contribute to the passage of electric current through an electrolyte solution. Migration. Ions movement across a potential gradient eliminate in voltmetry by adding a supporting electrode. Cyclic voltmetry. During the reduction process, the oxide species at the electrode surface is depleted and replaced by reduced species which diffuse into the solution. When the potential swap is reversed, the reduced species begin to reoxide back to oxidation as a potential approach to redox potential. This results in an increase in current in opposite, that is oxidizing direction, leading to an oxidation peak. The first figure is potential as a function of a time. Now the second figure is reversible cycle voltmegraph, and the third figure is cyclic voltmegraph for paracetamol. Now chronoamperometry. Instead of sweeping the potential, the latter is stepped in a square wave fashion to plot to potential just past where the peak should appear in a linear sweep voltmetry. This current then monitored as a function of time. Decay occurs because of the collapse. It is generally the function of a square. Here we can see the graph showing the square function. Amperometry, the analytical application of the chronoamperometric techniques. Now kinetic and catalytic effect. Catalytic cyclic voltmetry, like you can see there is a cyclic pattern for the figure for the A substance as well as the B substance, whereas the B substance has the high value of current with respect to the A. Now conductivity. The conductivity varies according to the charge on the ion, the mobility of ion and the degree of dissociation of the ion. The conductance is measured with the alternating current, and varying the frequency reveals admittance, which depend on the conductance, capacitance, and inductance. The component can be analyzed and displayed on an Argand diagram as admittance spectra. Field effect transistor. FET. Semiconductor. There are the two types of semiconductor, P types and N types. P type is basically hole, whereas the N type is basically the electron majority. Metal insulator semiconductor. With no potential, forming lever is aligned across the metal and semiconductor. For negative potential, electron accumulated at the metal insulator interface, while holes accumulate the semiconductor insulator interface. Energy level in the semiconductor bends upward. The potential needed to cause inversion is called the threshold potential. Semiconductor solution contact. When an N-type semiconductor contact a solution with the redox reaction, the forming level aligned with the redox potential. If the forming level of the semiconductor is higher, the electron flow into the solution, bending the conduction, and the valence bends upward. Field effect transistor effect. Monitor and control changes in MIC. Here you can see the N-types. In first figure we can see the p-type silicon, whereas we also see there is the voltmetry as well, and there are the schematic diagrams on downside. Modified electrodes, thin film electrode and the screen printed electrode. The first figure is the screen printed electrode, whereas the second figure is the schematic of the extra biosensor disposable electrode strip. Photometric sensor. Most bioagents were originally of the photometric types, such as they made use of change in a specific which involve a strong change in a photometric property. Optical technique, ultraviolet visual absorption, Fresnel's emission, bioluminescence, chemical luminescence, internal reflection spectroscopy, laser light scattering method. Some of the applications for the photometric sensor are optical sensor can be used for the measurement of the pH, whereas oxygen, carbon dioxide, and ammonia as well as iron. Ultraviolet and visual absorption spectroscopy. The electronic transition of organic molecules and the biomolecules occur in the ultraviolet visual regions of the electromagnetic spectra. Such spectra can be supplied for some information about the molecular structure and quantitative analysis. Thank you. That's all.